Hey everybody. This is our last lecture, at least the last one of my doing for this 703 term. Next you'll see Aviv or Gev for a number of online lectures. And so we're going to continue our final discussion on movement of DNA between bacteria. We talked about transposition. Transposition means essentially the hopping of a mobile genetic element that sometimes disrupts or integrates into target genes. We had transduction, which is the movement of DNA using phage, so phage infection of target cells. And in transduction, we talked about the accidental packaging of bacterial DNA by a P1 phage and movement of that bacterial DNA into a target cell where it can undergo homologous recombination with the genome in the target bacteria. In that context, we talked about using TN5 and kanamycin resistance as a way of mapping genes. Here we're going to talk about conjugation. And conjugation is a natural process that occurs between bacteria. It's a way of bacteria to transmit information from cell to cell. So bacteria obviously don't mate with other bacteria, but they do exchange DNA continuously. One of the ways they do that is via phage transduction that we talked about. But another way is conjugation. Conjugation here refers to essentially a bridging of one bacteria to another, where we have the movement of plasmids. All right, so what is a plasmid? A plasmid is a piece of episomal DNA And what do I mean by episomal DNA? Episomal DNA is DNA that's not in the bacterial genome. So it's maintained outside of the genome. It's self-maintaining. It has an origin of replication. We'll talk about it in a second. And this piece of episomal DNA can be transmitted from one cell to another. So what does a plasmid look like? Well, plasmid is a circular piece of DNA. Many of you have actually worked with plasmids before. They're really our working course in molecular biology. They're where we clone DNA, where we actually use these vectors actually make lots of DNA, so we can actually amplify and make gram quantities of DNA from very small concentrations when we actually want to use DNA to do other things. And so we use these for our purposes. Bacteria use plasmids, again, to exchange information, including exchanging drug resistance genes. And this is really a cause of significant human health concern because bacteria can use plasmid to not only carry single drug resistance genes, but many drug resistance genes, leading to plasmids that can lead to potent multi-drug resistance in the clinic. And so what does a plasmid look like? Well, a plasmid is a piece of DNA that has an origin of replication An origin replication simply allows the plasmid to be copied. So you can make lots of copies of a plasmid in a particular cell, and so it's self-sustaining. Unlike a virus that needs to integrate or it needs to undergo a lytic phase where it's like making a lot more of itself, a plasmid can sit outside of a bacterial chromosome for a long period of time and essentially tread water along with the bacteria replicating, so it can sustain itself for a long period of time. A plasmid has an origin of transfer And this is the mechanism by which it's actually going to move from one bacteria to another. So it's actually going to start at this little arrow point and move directionally, bringing its entire genome into a target cell. Another core component is a selectable marker, which is something like antibiotic resistance, so say ampicillin resistance, for example. So there can be a single drug resistance gene. There can be multiple drug resistance genes, again, which is really scary in a clinical setting. Many of these drug resistance genes are in transposons. So they were stuck into a plasmid by a hopping of a transposon from the bacterial chromosome or from another plasmid. So again, all of these movements between transposons and plasmids and viruses are really integrated into a network of ways that DNA moves around within a bacterial cell. A plasmid also encodes a number of transfer genes, and these transfer genes encode material necessary for this conjugation event to actually occur. So what does that look like? Well, if we envision a bacteria cell, so this is bacteria A, that has a normal chromosome, so this bacteria can also have a plasmid. So this is our plasmid. And plasmids are of varying sizes. They can be 50 to perhaps 100 kilobases in size. So they're still significantly larger than, or significantly smaller than the bacterial chromosome. And when bacteria A comes in contact with bacteria B, it can actually transfer the plasmid using a structure called a pilus. And this is essentially the conjugation event. The pilus, or the protein components of the pilus, are encoded by these transfer genes, which encode proteins that are necessary for the movement of a plasmid DNA from one cell to another. So when does this happen? So it happens when bacteria B doesn't have a plasmid. So bacteria B obviously has its E. coli chromosome, but it is negative for a plasmid. So a bacteria that actually contains a plasmid, and here we're referring to these as F plasmids, so is termed F plus. So an F plus strain is a strain with an F plasmid. And an F minus strain is a strain without an F plasmid. So if you mix F plus bacteria with F minus bacteria, roughly 1 in 10 F minus bacteria will become F plus. So importantly, F minus will become F plus, but F plus doesn't become F minus. So because the plasmid actually replicates in a cell, you have perhaps lots of copies, and we've engineered plasmids to be high copy plasmids so that we can actually encode for the presence of lots of pieces of DNA that we're interested in. Because it has lots and lots of plasmid, it's not giving all of its plasmids into the target cell, it's just sending one down there. So F minus becomes F plus, but F plus stays F plus. So what is the fate of a plasmid once it gets transferred? So most of the sequence of a plasmid is actually unique to the plasmid. Last time we talked about accidental packaging of DNA into a phage particle. And in that context, all of the DNA that moves over, except for the TN5 sequence, is actually just normal bacterial sequence. And so you can have homologous recombination between any sequence and any sequence. So any sequence that's brought in by P1 and any sequence in the bacterial genome, because they're essentially the same. In the context of a plasmid, that's not true. So say we have a plasmid, and for simplicity purposes, I'm just going to draw it here with the origin of transfer as this arrow. So the other element that is relevant here is what's called an insertion sequence. So an IS, or insertion sequence. And so this is a small sequence that has that shares identity with the bacterial chromosome. So the sequence shares identity with the bacterial chromosome, right? So let me redraw this, give myself a little space here. When you have a short sequence that has identity to a bacterial chromosome, so this is the bacterial chromosome, you can have recombination. So the recombination here is occurring in a very specific place. So it's occurring at this insertion sequence. And you have a recombination event there. And what that recombination event does is it actually brings the whole piece of plasmid DNA into the target bacterial chromosome. So you can imagine a root extending here, going throughout the bacterial sequence, back down into the insertion sequence, and into the bacterial chromosome. It's a little like a loop of thread. If you just pull that loop of thread out, then you actually get a straight piece, and that straight piece actually inverts the orientation, or the apparent orientation, of this origin of transfer. So what you get is a piece of bacterial chromosome that looks like this. So these two species can essentially interconvert. They can go back and forth and back and forth. How does this flip out? Well, think of it this way. So we have a looping piece of bacterial chromosome where this at any given point can recircularize and pop back out of the genome. So these two states, as I mentioned before, this is referred to as an F plus state. So a state where you have a bacterial chromosome and you have an F plasmid. And here at the bottom, we have what's called an HFR state. HFR stands for high frequency recombination. And in the HFR state, again, the whole plasmid sequence is now integrated into the bacterial chromosome. So this represents a really interesting state for a bacteria because of the presence of this origin of transfer. So a typical plasmid that has a normal origin of transfer, when it goes from one cell to another, it has a break that's essentially induced at the point of this origin of transfer. So the tip of this arrowhead is where you actually have the cut of DNA, and then you actually have a transfer of DNA from one cell to another that starts from the tip of this arrow. So the arrow leads the way. Everything behind this arrow gets taken in the order that it occurs behind this arrow. In the case of a plasmid, this transfer is very rapid because it's a small piece of DNA. So let's think about what happens in the context of a HFR state. So in the HFR state, we have our inserted plasmid, our two insertion sites. So we have two insertion sites because we had an insertion site in the plasmid and we had an insertion site in the bacterial chromosome. And essentially, half of this insertion site came from, each came from the plasmid and from the bacterial chromosome. The recombination can occur at any site within this insertion site, and it's impossible to tell the difference between them. But again, we have two because we had two to begin with. We had one in the plasmid, one in the bacterial chromosome. So there's no gain or loss of genetic material here. We're simply seeing both of these now appear within this inserted HFR state. So let's put in some genes. So say there's gene A there, and there's gene B there, and gene C there. So in this HFR state, believe it or not, you can still actually have transfer of genetic material from one cell to the other, except in this case, you're not just transferring the plasmid, you're actually transferring the whole bacterial chromosome. And so what is attempted, or what during conjugation, is to actually start bringing in this material, again, starting at this arrow point. So you'll have a break, and you'll have the transfer of DNA starting at the arrow, going through A, and then the rest of the bacterial chromosome, and then going to C and B. So in an HFR state, you can have transfer of the whole bacterial chromosome. This takes a long time. The whole bacterial chromosome is about 5 megabases, or 4.6 times 10 to the 6th in E. coli. But this transfer, I said whale instead of whole, okay. Transfer of the whole bacterial chromosome, it takes about 100 minutes to move the whole thing across. And the timing of transfer varies considerably depending upon where a gene is within the bacterial chromosome. So here, A is transferred first because the point starts transfer and everything else follows the arrow. So A is transferred first, then C and B about 100 minutes later. So some markers are transferred early, others are transferred late. Now this is distinct again from the F plus state because in the F plus state, no bacterial genes or markers are transferred. So again, in the context of just the plasmid, you're just transferring, or the F plus state where the plasmid is sitting episomally outside the bacterial chromosome, you just have a transfer of this, of the plasmid itself and not a transfer of any chromosome markers. But once the plasmid actually gets inserted into the target genome, then it carries all of these bacterial loci, all of these markers along with it, and it will attempt to transfer the entire bacterial chromosome. Okay, so now that I've magically cleared the board, let's redraw our HFR string. So we had a gene A, then we had our inserted plasmid, then we had B and C. Again, this is an HFR. All right, well, so what happens if we actually have another insertion site somewhere else in this bacterial chromosome? So let's put one down here, downstream of C. So we have C, then we have insertion site, and then we have D, another gene of interest. So you can go back and forth popping this plasmid in and out. So you can go back and forth from this F plus state to the HFR state. But you can also have a different recombination event. So say instead of having a recombination event between here and here, which leads back to the F plus state, you actually have a recombination that occurs between here and another insertion site. So this one downstream of C. So what does that look like? Well, we can actually draw it as a recombination event. So we are looping around. We have our origin of transfer that is flipped in the other direction. We now have another insertion site, so this insertion site. Then we have B, and C, and D. So essentially, I've just flipped this sequence around, so this insertion site is now lined up with that insertion site. So A is there, D is there, and then B and C are looped together within another piece of DNA that can excise out of the genome. And so what are we left with? Well, we're left with A and D, and then we're left with a plasmid that has an origin of transfer. It has the B gene and the C gene. So we're going to call this an F prime strain, a strain that has what's now called an F prime plasmid. So an F prime plasmid contains genomic DNA. So it contains this B and this C gene. Now you have a piece of the bacterial chromosome that actually has been transferred into this plasmid, and this plasmid can then transfer DNA from one cell to another. So this is a way that bacteria is using to actually communicate its own genomic DNA into a target cell. Of course, it can do that in the case of the HFR, but in the case of the F prime, it's doing it a little bit differently. So an F prime strain transfers chromosomal DNA, but a little bit differently than the HFR strain, because some genes are transferred quickly. And so here, imagine that B and C are going to go over quickly, and some genes are not transferred at all. So in the HFR strain, we're transferring A, all of the other genes, D, C, and B, all of them go together. Some are transferred early, like A is transferred early, and B and C are transferred, but B and C are transferred very late. In the case of an F prime strain, the markers that are transferred, the genes that are transferred quickly, but many or most are not transferred at all. Another important distinction is you'll see that in the case of B and C, so B and C were the last to be transferred in the HFR strain, but they're transferred early in the F prime strain. So genes that are transferred late in the HFR are transferred early in F prime. Another thing you might see is that there is an orientation of these insertion sequences, and so you could draw them functionally as like this. They're just DNA sequences, and so they have an orientation, 5' to 3'. And so when you have this looping over of DNA, you're aligning the orientation of these insertion sequences. It's an interesting thing for you maybe to think about just as an exercise. what would happen if you actually had insertion sequences that were in the opposite orientation. So they were sort of palindromic to one another. And the short answer is that instead of having something looped out, you'll essentially have something inverted. And so in this case, it would look like if they were inverted. So here you would simply flip the orientation. And so there are lots of different ways where plasmids and integration of plasmids can not only move DNA from one cell to another, but the insertion sites can actually recombine to produce interesting different orientations of genetic material in the cell. And so how can we actually use this conjugation for experimental purposes? So one of the things that we can do is we can actually use this to introduce new genetic material into a target cell. And so say we have a strain that is lac minus, and we look at growth on lactose. A lac minus strain can't grow on lactose. A lac plus strain can obviously grow on lactose. But what if we actually introduce a plasmid that has a gene essentially for gene essential for lactose production into this lac minus strain, and we can draw that as lac minus and F prime lac plus. So this essentially, if we introduce a plasmid that contains an essential gene that's missing in the lac minus strain, we can actually restore production of, or the ability to grow on lactose as a food source. And so what we're doing here is we're constructing what's called a marrow diploid. And a marrow diploid is essentially a partial diploid for genes on the F prime plasmid. So it's a way of introducing a diploid state. And so in doing this, we can tell, for example, that lac minus is recessive. And as you know, we can only determine dominant and recessive if we actually have two different alleles. So if we introduce a wild-type version on a deficient background, we can introduce two alleles, and we can determine whether this allele is dominant or recessive. And in this case, it's recessive. We can also introduce, say we have two genes that are next to one another, and so say we have a bacterial strain that is A minus B plus, and we have a F prime strain that is A plus B minus. We're actually introducing a wild type version of the A gene and a mutant version of B gene onto a background that is deficient for A and proficient for B. In this case, this is essentially a complementation assay. Again, something that is really only feasible in the context of this marrow diploid, where we're using plasmids to move around this DNA. So importantly, we can also use this kind of approach to look at or to identify gene deficiencies or to try to complement gene deficiencies that exist in a bacterial strain. So say, for example, you have a mutation in bacteria that is not allowing for growth on lactose. You can generate a whole library of different plasmids that all contain a little piece of DNA and look for pieces of DNA that can then restore the ability to grow on lactose. So try to identify plasmids that actually contain lac genes. This is another kind of complementation assay, and one that we use very frequently to try to identify what is the source of a deficiency, can we actually correct it. So these kinds of approaches are used very frequently as tools to identify and move around genes of interest, to assess dominant recessive, to do complementation assays. We use plasmids again all the time to actually clone pieces of DNA, so trap them and generate lots of them so that we can actually use them using all the molecular biology techniques that we need. If we have a gene that we want to make a protein from, we actually clone it into a plasmid for manufacturing purposes. So plasmids are really fundamental in our ability to do all the molecular biology that we're interested in doing. This movement of DNA in plasmids, again, also is problematic because it is a mechanism by which bacteria actually acquire drug resistance mutations and transfer them from cell to cell, which underlies the etiology of a lot of drug resistance in really hardy or drug resistant strains that we use or we see in the clinic, or that arise via communication between bacteria in the clinical setting. So that's it in the context of DNA movement within bacteria. Next, you will hear or start to hear a number of lectures from Aviv, and so I wish you a great rest of the term, and be well.